And welcome back out. In this video, we will see how we can utilize the HHPRED tool, that is available through MP Bioinformatics Toolkit, and if for the analysis of protein. MP Bioinformatics Toolkit has a different suite of tools that can be utilized for alignment, sequence analysis, secondary structure, 3D structure analysis, classification, etc. So in this video, we are going to utilize the HHPRED tool that will allow us to predict a 3D structure of a particular protein. But first thing you need to understand is that HHPRED is basically utilizing a model on the backend, and it utilizes the same pipeline of analysis as a modeler tool. But in this case, it is a entirely online available toolkit that you do not need to worry about anything else, such as template finding or something like that. So you just need to provide in the protein sequence, of the target protein, and it will do that as for you. But you obviously have to select a particular template at the end for the dedication of the template against target alignment. So what I'm going to do is I'm going to utilize this dead protein. It literally means dead. So it's a death factor domain containing protein. It does not really have a structure. So we are going to utilize this protein for the further analysis and prediction of protein structure. So let's go to format, fast canonical, and then select the protein sequence. Let's copy it. Go back to our HHPRED tool. You can find this link within the transcription of this video. Just input this sequence over here, and it has understood that it's supporting FastJ, and then we can move on. You can change the parameters. In most cases, please keep it at default unless you know what you are doing. And after that, just go back to input. If you need to align two sequences or produce a multiple sequence alignment, you can do that as well. But in this case, we do not need to do that. So just select particular database, because you have to select a database. Otherwise, we cannot really do a template. We cannot really find a template, and we cannot really do homology modeling. So we need to define a database. In most cases, all of these are protein databases, and they are defined protein databases. MMCF PDB is the default, which is updated to the July month, and you can select other databases such as PFAM, Uniprot, Swiss for viral, as well as COG can be selected, and SBA conserved domain can be selected, Smart can be selected, Tiger from the announcement selected. So I'm just going to keep the default. Then we have to select the proteome. So this is a very unique feature of HHPRED. If you know that the proteome that you're trying to analyze is actually of a particular species. For example, the protein that you actually are trying to predict, it belongs to the proteome of human, since it is a human protein. Let's go back. Since it is a human protein, so we know that it is a human protein, so we are going to select the human. That's this fine human protein. Okay, so there's no. The name protein is fine. I'm sorry, it is a scientific name, so it is Homo sapiens. So I'm just going to select Homo sapiens. And in case, let's suppose that you have a rat or you have other species and you would like to analyze that. In that case, we have to select the particular proteome of the particular species. So if you recall the PDB video, in that video, as I talked about how the PDB sources were classified within the PDB and protein data bank, and that they can be utilized for finding out a particular protein that resides within the particular species. So this is the same scenario. We are actually classifying that information. We are selecting that particular information. So I'm just going to click on submit, and it will take some time before the results are provided back to us. And once the results are ready, we will then analyze them further. As you can see, here is seeing the protein sequence, predicting sequence features, and it will take some time before the actual analysis is completed. But the best thing about this is that we can utilize this tool rather than having to install another tool on our computer and all of that. But there are some very unique features about this, which we will keep discussing as the video goes on. Okay, so it took some time, but our results are back. As soon as you Joe by day that green color, the results are ready to be analyzed. So there are various things that you need to actually understand what actually happens after the results are ready. Right now, your protein has not been predicted. Your protein searches has not been predicted. It is just analyzed through various databases searches, such as Uniport for the database that you selected. The results are ready, and the prediction is ready. So what it really does is that this is your query from first to 318 amino. So let's verify that our protein is indeed 318. There we go. It is 318. Let's go back and just generate it. If we can move around, just let's let this one, we can move now. But after that, we have to click on some section. I'm not going to do that. So these are all the proteins that it found and to be matching. If you hover over them, you can see probability score, E-score, XP, and the accession ID provided and predicted information. So all of them would be obviously isoforms, and it found them to be actually correlating with them. They are different accession, but same protein. And then we have the domain and exact domain information as well. For example, these are some proteins that have the, maybe this is the region where the protein domain is available. So that is why all of these proteins seem to have some sort of correlation over this region. So let's go now, and these are the region. Basically, the darker the color, the reddish the color, the more identity it has as compared to the lighter color. For example, green, blue, and that. So after that, all of these results are available in a table manner. So we can select the top 10. We don't need that 25. Then we can see, okay, so this is the same protein. This is also the same protein. This is also the same protein. Same protein, same protein, same protein. So we cannot really utilize them for the analysis for further because all of them are the same protein. How can we actually predict the protein structure if they do not also have protein structure, because all of these are the same isoforms from this protein, so they obviously they do not have protein structures. How can we utilize them for the protein structure? So these are basically secondary structure analysis, and this is a better blast analysis as compared to an analysis that is performed by a blast or NCBI uniform, because it does not provide us secondary structure analysis. So HHPRED actually requires the secondary structure analysis and the blast analysis. So it provides you the exact region of the coils, helices, beta sheets, and then it also provides you the aligned regions. So these are all the results that we can utilize and for further analysis. But since, as I said earlier, that all of them are the same protein, that they may not have protein structure. So let's go back to a table, and this time we can let's utilize this one. DNA binding that I think this is the same one as that. And this is the same one as it. So let's select more entries. And this time we can select astratic phosphoprotein. This seems good, but it doesn't really have length. So what I'm going to do is I'm going to create a multiple selection. And let's see what really happens. I'm just going to select the 10. Let's select top 10 sequences and we will move on only. After the selection, what you really have to do is after the selection, what you really have to do is you have to go to the bottom of this analysis and just make sure all of them, if you need to visualize them. After that, once you have made your selection, once you have understood which proteins you would like to utilize, just go to forward, click on forward, alignment view. You can view this in alignment view, you can view this in the HHBlast, HHOMP. These are our different tools. Otherwise, you can just click on model using selection. It will create a model according to the selection you made. So let's click on model using selection. And then it will take some time. The submission will be submitted again. It will take some time, and then would provide us the protein structure if there are no errors. Since there are errors, select templates field 2% checks only pretty entries may be selected as well. So it's a very good thing that actually selected them, because it provides you an idea that if the protein structures are not available of a particular protein that you selected as a template, then it will provide you an error. So let's go back and let's try to select a particular protein that actually has a protein structure. So this is actually a problem within this tool, that it does not really allow you to understand which protein sequence has a structure that can be utilized for that structure prediction. So I'm going to select this particular protein structure that has a protein structure available. Okay, so the better option right now is that what we really need to do is, because all of these sequences are protein sequences, and all of these entries basically are, or hits are protein sequences, and they do not really have protein structure available on the PDB. So we cannot really obviously use them for the protein structure prediction. So what I'm going to do is I'm going to enter PDB within the search bar, and it will provide me those entries, those hits that actually have a protein structure available on the PDB, so that we can utilize them as a template, because they were actually available. They were actually available within the hit list. So we are actually filtering out the PDB structures that can be utilized for the structure prediction. So probability is 99.5, the top one. And the length is 91, which is not as effective because our protein is of 318 length. So what in this case, what you can do is you can select the biggest protein, and if there is available, and for example, by doing that, you can, since this is an interactive table, you can filter out the best protein that is okay, but you have to consider the probability as well. So we can find a protein that has some sort of wood length. Let's utilize these four actually. Okay, so we have selected this as templates. All we really need to do is go back to our top of the page and then click on model using selection. And now these four different protein structures will be utilized as a protein structure. This seems to win her. Again, let's go back. Maybe I selected some different protein. Okay, so just go back to target length. Okay, just select this one. I'm gonna go to new protein and go back to the top, model using selection. There we go. So this protein was selected as now there is a node and error, and as you can see the alignment has been produced, and now we can forward it to modeler, and modeler will take some time and it will produce such protein structure. But first of all, we have to provide in the key, which is model. And just click on submit, and it will take some time. Submission is given and it will start producing that protein structure. So our protein structure is available. Just click on download PDB file, and this is our model. It will also display over here. So it is actually quite efficient. As once we have started practicing with this tool, it is quite efficient. The protein structure building is the instant. As you see, it not really take any time, but the template section is some sort of an issue with this tool. That the prediction is done. So this is the only region that actually was aligned against the template, and not the entire region, because we did not actually find a protein that was actually a wood protein that could be in that. So this is a protein file that we downloaded and can be utilized for further analysis. So that was all. I hope you understand this video. We will actually be analyzing this toolkit in further for sequence analysis, research analysis, classification as well. So you should be good familiar at you should really become very familiar with this tool at the end of this course. So that was all. I hope you understand this video. If you have any question, you can reach us and we will most of the year. Thank you so much. Have a great day.